Konnichiwa guys, welcome to our channel Loops of Biology. I'm Yogiraj Jakkal taking you all to the one shot of Biomolecule. As I have said, this is a summarized lecture for a chapter of Biomolecules. In this particular part, we will be covering your topics such as Introduction to Biomolecules, Carbohydrates, Lipids, Proteins, Nucleic Acids, Enzymes and Secondary Metabolites. However, the proteins, nucleic acid, enzyme and carbohydrate. These are one of the most important topics if we are talking about biomolecules. Let's move on to the introduction. In the case of biomolecules, I am able to see carbohydrates, which are also called as polysaccharide in its polymeric form. If I'm talking about lipids, there is a complex structure called as triglycerides. If I'm talking about protein, there will be your polypeptide. While if I'm talking about nucleic acid, there will be polynucleotide. So there will be polynucleotide. These are one of the complex structures which are formed whenever I'm talking about carbohydrate, lipids, proteins and nucleic acid, and all components are basically integral component of biological system. When we are talking about biomolecules, we are studying the field called as biochemistry because we are integrating the part of chemistry inside the biology. That's why the stream is basically called as biochemistry. If we are moving on to the first question, the most abundant element present in the plant is, whenever we are talking about any abundant element, the element should be prime element. The element has to be prime element. And as we are talking about plants, animals or any organism, all organisms contain organic compounds. And in every organic compound, you must be knowing that the carbon is going to be the most abundant. So there is going to be, the answer for this question should be carbon. Got my point, guys? Let's move on to the next question. The major portion of the dry weight of plants comprise of, whenever I'm talking about dry weight, all right, the dry weight is nothing but when the water is removed out. And whenever I am removing the water, I am left with only organic content. So wherever I am going to see the carbon, that will be my answer. So options 4 and 3 are wrong. Next case, if I'm talking about carbon, so alongside there should be a presence of oxygen as well. The oxygen is still present. With carbon atom. So whenever I am going to see oxygen carbon combo, that will be my correct answer. So option 2 will be the correct option for this particular question. Let's move on to the first biomolecule, guys. That is nothing but our carbohydrates. So whenever I'm talking about carbohydrate, the monomeric unit is nothing but your simplest unit of carbohydrate. Whenever I am talking about simplest unit, that is nothing but called as monosaccharide. And this monosaccharide is divided based on functional group and based on the number of carbon atoms. So if I'm talking about the monosaccharide, that is belonging to case aldoses. In this particular case, the functional group is aldehyde. So aldehyde is nothing but your main functional group. That if I am talking about ketose, that is nothing but your ketone as main functional group. So there are two categories of your monosaccharide, that is nothing but aldoses and ketoses based on a functional group of aldehyde as well as ketone. Whenever I am going to add this particular monosaccharide together through anabolic pathway. So whenever monosaccharides are undergoing anabolism, so they are following anabolic pathway. So during that anabolic pathway, I'm going to form my polysaccharide. And for joining any monosaccharide, there should be some bond. Suppose this is my monosaccharide of sugar and this is joining to another monosaccharide A dash. So there is a bond formed by removal of water and it look like an ether. And it look like an ether bond. And this is basically called as glycosidic bond. Glycosidic bond. Next case, during that bond formation, there is always the removal of water molecule is going to take place, and the general formula for carbohydrates is nothing but CXH2O and Y. This is basically X and Y and number. So that is completely based on number of carbon and number of water molecule present inside that particular structure. So they are not as such water molecule, but we can also write this particular formula as for monosaccharide CnH2nO. This formula works when it comes to monosaccharide. So to understand whether the molecule is monosaccharide or a complex carbohydrate, for complex you should follow this one, and for monosaccharide you should follow CnH2nOH. There will be no problem. So now based on carbons, if I am talking about the division of monosaccharide, that is completely based from carbon number 3 chain till carbon number 7. So if I am talking about 3 carbon, there will be triosis. 3 carbon is triosis. 4 carbon is called as tetrosis. 5 carbon is pentosis. 6 carbon hexosis. And finally, 7 carbon is nothing but heptosis. So these are 5 different categories when we are talking about division of monosaccharide based on the number of carbon. So let's talk about the structure of glucose. If I'm talking about glucose, then I can see on the first carbon there is aldehyde. So glucose is belonging to aldoses category. Alright. Now, if I'm talking about aldoses, along with that, there are total 6 number of carbon. So this belongs to a case of hexosis. So based on carbon number, it is 6 carbon. It is 6 carbon compound with RCHO group present inside it. So if I'm talking about glucose, on that particular structure, on the carbon number 3, there is a change in the position of hydroxyl group. Every hydroxyl group in the glucose is present on the right hand side, if you can see. But on the third carbon, it is present on left hand side. And this is basically called as Fischer projection. It is called as Fischer projection structure of glucose. That is nothing but straight chain structure. Straight chain structure of glucose. But when I am going to see a stabilized structure, whenever I am keeping the structure in straight chain, that is not going to be a stable structure as such. So whenever I am going to put that into a cyclic form, so that particular structure is called as Haworth projection structure. And this Haworth projection is nothing but your stable structure, which is formed based on placing of hydroxyl group above or below the plane, above or below the plane. So that's why this structure is nothing but completely based on the planar symmetry. Alright, so whenever I'm talking about OH group, on the first carbon of the cyclic chain, guys, first carbon of the cyclic chain. So that is called as anomeric carbon. So the first carbon of cyclic chain is nothing but my anomeric carbon. So whenever on this anomeric carbon, the hydroxyl group is present below the plane. The hydroxyl group is present below the plane. That particular structure of glucose is called as alpha glucose. Alright. So whenever I'm talking about the first carbon of cyclic chain, this is nothing but my cyclic chain. So in this cyclic chain, whenever I'm talking about my first carbon, the hydroxyl group present on this carbon is going to decide whether the glucose is going to be alpha or it is going to be beta. If the hydroxyl group present is below the plane, then it is going to be alpha glucose, and if that is present above the plane, then that will be your beta glucose. So for alpha glucose, OH below, and for beta glucose, OH above. Only the position of OH on first carbon is changed, rest the structure remains same. Got my point, guys? This is nothing but your structure of glucose. So let's talk about epimers. Whenever I'm calling epimers, so epimers are nothing but with respect to the standard structure, if the position of one functional group is changed either above or below the plane, then that particular new structure is called as epimer. Let's see what does that mean. If I'm taking glucose as my standard structure, so this is kept standard. So whenever I'm talking about glucose as a standard structure, then I am well versed that on the third carbon, the OH position is always has to be on the left side. So the new structures are also going to have the same criteria. Alright. So in this case, if I'm talking about mannose, so mannose is basically showing C2 epimer. Now what do you mean by C2 epimer? That on the second carbon, the hydroxyl group which is present on right hand side, that will move on left hand side of the structure. So can you see this? This is carbon number 1, this is carbon number 2. So on the second carbon, I am keeping my hydroxyl group on the left hand side. Rest the structure of glucose remains same. Alright. So in this case, the hydroxyl group of second carbon is changed. Alright. While if I'm talking about the galactose is nothing but my C4 epimer of glucose. And whenever I'm saying C4 epimer, that means keeping everything same but changing the OH position on the fourth carbon. So this is 1, 2, 3 and 4 carbons are going to be on that particular fourth carbon. I am going to change my hydroxyl group position. Alright. Along with this epimer, there has to be some chiral carbon atom count. Whenever I'm talking about chiral carbon, this means one carbon atom is attached to 4 different groups. Alright, one carbon atom is attached to 4 different groups. So if I'm talking about in every of this structure, I can see that this carbon, the carbon number 2, 3, 4 and 5 are nothing but my chiral carbon atom, and they are basically represented by asterisk mark. Alright guys, so this asterisk mark is going to show me the chiral carbon atoms which are bounded by 4 different groups, and that keeps the particular structure in a optical isomeric form. Alright, so these are basically, whenever the chiral carbon atom are there, they are optically active. Optically active. Alright, so this is a part of epimer. Let's move on to the next part, that is reducing and non-reducing sugar. When I'm talking about reducing sugar or non-reducing sugar, that is completely based on the aldehyde, and ketone are present in a free state. That particular structure is called as my reducing sugar. But when this aldehyde and ketone are undergoing the bonded condition, that is nothing but your case of non-reducing sugar. If I am not having a Fischer projection structure, then how can I decide whether aldehyde and ketones are in bounded or non-bounded structure? That's my question. That should arise in your mind first. Then the next definition for this will be when the OH group is free on anomeric carbon, then the structure is reducing sugar. Otherwise it will be a non-reducing sugar. Let's see what will happen. As I have told you, the anomeric carbon is nothing but the first carbon on the cyclic chain of a structure. So I can see that in case of this particular structure, this is my first carbon, while in another, this is my first carbon. Alright, so in this lactose, there is beta galactose and beta glucose is used. Whenever I'm talking about beta galactose, this is my structure for galactose, guys, and this one is a structure of glucose. All right, you can see in beta the OH position is above the plane. All right, and this bond which is formed is nothing but my glycosidic bond. So in this structure, I can see that there is a free hydroxyl group present on anomeric carbon. That's why it belongs to the reducing sugar. So there is in beta glucose on first carbon of cyclic chain, OH is free. But when we are talking about the structure of sucrose, that is known as table sugar or cane sugar, you can call this as cane sugar. The sugar present inside a sugar cane. So in that case, I am going to see glucose and fructose as my monomeric component. Now in that particular case, I can see that the first carbon of alpha glucose is bounded with the second carbon of beta fructose. Alright, so the first carbon of alpha glucose is bounded with the second carbon of beta fructose, and during that particular case, the second carbon acts as an anomeric carbon because this is first inside a cyclic structure. So in fructose, second carbon act as first carbon of cyclic structure, and that's why there is no anomeric carbon with free hydroxyl group. So there is no free hydroxyl group on anomeric carbon, and that's why your sucrose belongs to non-reducing sugar. All right, so let's move on to the next part, that is your question. Which of the following is a non-reducing sugar carbohydrate? As we have talked about it, that is nothing but whenever there is no free hydroxyl group on an anomeric carbon. I hope everyone is clear about anomeric carbon. If anyone has any doubt, please write it down into the comment box so that I can give a proper response with a proper, we can say, logic and the concepts behind it. All right. So which one of the following is non-reducing carbohydrate? So that is nothing but here sucrose, which is formed by alpha glucose, and there is going to be beta fructose. All right. If I'm talking about ribose 5-phosphate, this is your 5 carbon monosaccharide, which is belonging to a case of pentoses. If I'm talking about lactose, this is beta galactose and beta glucose is going to be used. And if I'm talking about maltose, that is nothing but your alpha glucose combination. Alpha glucose combination. This 3 belongs to disaccharide. This 3 belongs to disaccharides, while this pentoses belongs to your monosaccharide case. Alright, so disaccharide and monosaccharide. Mostly monosaccharides are not going to be your non-reducing carbohydrate. Let's move on to the next, that is lactose is composed of, right now I told you, that is glucose and galactose. So option number 4. In 1998 it has been asked, and there are many of the questions which I am going to cover which are asked in the NEET examination as well, and those are very tough. If I am moving to the next part, that is Benedict solution. So to understand whether the particular compound is having reducing sugar or not, there is a one solution test called as Benedict solution. So this analyzes the sample containing reducing or non-reducing or non-reducing sugar. Now, as we have talking about reducing sugar, that means it has to reduce something. And what do you mean by reduction is nothing but reduce the oxidation number. Reduce the oxidation number, or add H plus, or remove that is oxygen. So there is oxygen is removed. So in this particular case, whenever I'm talking about Benedict solution, this is containing copper 2 plus ions. Alright, so whenever there is a presence of reducing sugar, if there is presence of reducing sugar, this copper 2 plus ions are converted into copper 1 plus ions. That means from the plus 2 oxidation state, it is moving to your plus 1 oxidation state. So this is nothing but reduction of that particular solution. So this solution, first of all, look blue in the color, and whenever based on the concentration of reducing sugar, based on a concentration, so this changes, all the changes are based on concentration of reducing sugar in the sample, So based on the concentration of reducing sugar, the color of this particular solution is going to change to light green, then a dark green, then yellow, then finally you are going to see orange, and if there is a high level of reducing sugar, then that will be taking place into the brick red solution. So this is basically used to analyze urine sample, urine sample. All right. So this is all about Benedict solution. Let's move on to the next part, that is nothing but your polysaccharide. So as I have told you that every monosaccharide unit is bound together through a glycosidic bond. Every monosaccharide unit is bound together through a glycosidic bond. So whenever multiple monosaccharides are bound, they are going to form your polysaccharide. So in this polysaccharide, I can see a structure of starch, and this another structure is of glycogen. So as I have seen starch, the starch is basically a component which is formed within a plant. Alright, so during photosynthesis, there is a formation of glucose, and finally the glucose molecules are bound together to form a starch. So this takes place due to binding of glucose molecule. So this is take place of binding of glucose molecule during photosynthesis, and that too inside your case of only and only plants. Alright. And if I'm talking about glycogen, that is again a binding of glucose molecule take place. But these glucose molecules are basically excessive inside a blood. Whenever the blood glucose level is increased above its normal concentration, then that particular glucose is undergoing anabolic pathway and forming a polysaccharide. So this take place inside your liver of an organism. So this polysaccharide formation follows anabolic pathway. Alright. So if I'm talking about starch, so starch and glycogen are basically similar in their structure. So there has to be some similarity. Now, what is similarity? That means they are having straight chain structure as well as branches. So there is a similarity of having straight chain plus branched chain structure. But whenever there is a straight chain, the repeating units are differed in case of starch and glycogen. Now, what do you mean by repeating units? If suppose there is A bounded with A, another A, another A, another A, 5, 6, 7, 8 times, and after that there is going to be a first branch, then again the straight chain follows. So whenever after 7 to 8 units, first branched unit is seen, that case is nothing but of starch. But in case of glycogen, there are only 2 to 3 units are present, and again there is going to be a presence of branch. So the repeating units between intervals, interval of branch chain, those repeating unit number is going to get differ whenever I am considering that in the case of starch and glycogen. Alright guys, so whenever I'm talking about a straight chain, that is called as amylose, and this amylose is formed by alpha 1 to 4 linkage. That means first carbon of the alpha glucose to fourth carbon of the alpha glucose. That if I am considering a branched chain structure, that is bound by 1 to 6 linkage. So and this is called as amylopectin. So your starch and glycogen are basically formed by amylose and amylopectin. Together, amylopectin is a branched chain structure while amylose is a straight chain structure. Alright guys, so this is a part of your starch and glycogen. So in case of amylopectin, the first carbon of glucose is bounded with the sixth carbon of glucose. Got my point? Should we move on to the next part? That is nothing but your first question. Now, some of the concept I am going to cover inside the question itself. Macromolecule chitin. This chitin is basically used as exoskeleton in arthropoda phylum. So arthropods. So there are some organisms with a jointed appendages. Their exoskeleton, that means the covering, is basically made up of chitin, and that is also present in the cell wall of fungus. Cell wall of fungus is also going to contain your presence of chitin. Alright guys. So this chitin is made up of N-acetyl glucosamine. So now as we can see, N-acetyl glucosamine means there should be a presence of glucose. Alright. And this glucose units are repeating. So these are repeated. So if I'm talking about repeated glucose unit, that means you are going to see a case of polysaccharide. And there should be your nitrogen containing polysaccharide. As amine group is going to contain NH2, that means there is going to be the presence of nitrogen. So chitin is nitrogen containing polysaccharide with monomeric unit as N-acetyl glucosamine. This is also have been asked in the exam of NEET. Next case, cellulose, the most important constituent of plant cell wall, is made up of. If I'm talking about cellulose, the cellulose is again your polysaccharide that is made up of beta glucose units. Alright, so wherever I'm going to see beta glucose, that will be my first of all answer. So alpha is cutted out. Again there is alpha. So there is no alpha as such. So the answer is option number 3. But let's talk about cellulose a little bit more. If I'm talking about cellulose, that is made up of beta glucose. So this is forming unbranched structure with beta 1 to 4 linkage is going to be seen. If I'm talking about 2 units of cellulose, monomer. Alright, whenever I'm talking about 2 units of monomer, this structure is called as cellobiose. Alright, so if I'm taking 2 beta glucose and joining it together, it is forming cellobiose. If I'm taking more than 2 and joining it continuously in a straight chain structure without unbranching, alright, so there is going to be your case of cellulose structure. So in the cellulose structure, I am going to see beta glucose as the main component, and it is main constituent of the plant cell wall. So whenever you are going to eat any raw plant, so in our body in ancient time, there was a structure called as vermiform appendix, was very helpful for the digestion of cellulose because it will use to release cellulase enzyme. So for digestion of cellulose inside our body, there should be a need of vermiform appendix if I am going to eat the plant in its raw form, because cellulose has to get degraded, otherwise it won't be able to digest it inside our body. Alright, and that work was used to be done by vermiform appendix. Now it is a vestigial organ. I don't think anyone is going to eat the raw paper or pages of book as a food material, right? So let's move on to the next part, that is most abundant organic compound on earth. It is nothing but your cellulose, guys. It has been asked 2 times in a NEET, that is 2001 and 2004. The most abundant because there is plants. So based on number of plants, you are going to see that cellulose is nothing but most abundant organic compound. Let's move on to the significances and wrap up with this part of carbohydrate. Significances of carbohydrate is nothing but blood group. So based on different blood groups, that is nothing but here A, B, AB and O, the surface of RBCs, that is nothing but red blood cells are loaded with different combination of carbohydrate, and that is hydrates, and that is going to be deciding the type of blood group, whether it is A, B, AB or O. Now it is also acting as cell receptor, which is modified inside your Golgi apparatus. If someone must be knowing about Golgi apparatus are basically modifying proteins. So in the case of cell membrane, whenever the cell membrane is having a protein outside it, this is modified with this particular notch-like structure, and this notch is made up of carbohydrate, and that is called as cell surface receptor. All right. Next one, it is acting as energy source. That means it is going to provide me with ATP whenever the carbohydrate is undergoing catabolism. Next significance is the structural component. As we have seen that cellulose, chitin, one of the main structural component units of your carbohydrate. So there is cellulose, there is chitin, and this belongs to case of homopolymer or homopolysaccharide. But if I'm talking about hyaluronic acid, so that belongs to heteropolysaccharide, and this is present in the outer membrane of ovum, or you can say egg cell. Alright, in case of sperm and egg, the egg is basically having a presence of hyaluronic acid as one of the main components of the membranal part to be remain integrated. Next case is storing unit. In that particular case, you are going to see that the storing unit is basically done through anabolism, and these are basically stored inside a plant as starch, and if I'm talking about animals, mostly in the form of glycogen. So these are basically storing units. So whenever they required energy, they have to break that down and obtain the energy from those particular component, and that is also used inside a case of research, guys. All right, let's move on to the next biomolecules, that is nothing but your lipids. If I'm talking about lipids, the monomer is again going to be your fatty acids and glycerol. So here there are two monomers, but the complete structure of lipid is not in a repeating form. The lipids are not polymer. All right. Why not polymer? Because there is no repeating units. Only a glycerol, which is acting as a backbone of structure. On that glycerol, the fatty acids are came and attached directly through ester linkage. So this is a case of lipid, guys. So whenever I'm talking about lipid, a structure is comprised of fatty acids, glycerol, and those structure is basically formed because of ester bond. All right. In case of lipids, the hydrogen is to oxygen ratio is always more than 2:1. Please remember this because this is very, very important. If you know the structure of fatty acids and glycerol, you won't be able to memorize that particular ratio. All right. So in case of fatty acid, there are two cases, that is saturated and unsaturated fatty acids which are joined on the glycerol, which is 3-carbon structure. Alright, so belongs to your case of trioses. Let's see what does that come to. Whenever I'm talking about fatty acid, they are long chain of carbon and contain carboxylic acid as a main functional group, that is RCOOH. Glycerol act as a site of attachment. As I have told you that glycerol is a backbone of structure and it provides a site of attachment for this particular fatty acids. Both are joined together through a ester bond, that is nothing but RC double bond OO R dash bond. So this particular C double bond OOR, this is your ester bond which is formed. You can draw a structure and check for it. If not understood, please write it down in a comment box. I will give that particular explanation to you guys. The most common type of lipid is triglyceride, one of the important case because this is asked inside your NEET examination. Right? Which of the following is the most common type of lipid? Those are nothing but your case of triglyceride. Now, whenever I'm talking about tri, means there are 3 units. Is there 3 units of glycerol? No, because there are no repeating structure of your glycerol and lipids. But there is on one glycerol molecule, whenever I am going to add 3 fatty acid, this structure is called as triglyceride. Alright, and there are no repeating unit of a structure. That's why it does not consider to be a polymer. We have covered that up. So let's move on to the next part, that is saturated fatty acids. Whenever I am talking about saturated, that means it is going to have a presence of single bond between carbon to carbon. Then there is a chain starts from 4 carbon and they are solid at room temperature. One of the important point, guys, that they are solid at room temperature. Now they are most stable elements. So whenever I am talking about a structure, the structure is stable and less reactive if it doesn't contain double bond or triple bond. Alright, so as we have seen in the case of saturated fatty acid, there is only a presence of single bond between carbon to carbon atom, and that's why there is only a presence of sigma bond, and they are basically more stable and less reactive. For example of this particular saturated fatty acid, you can consider palmitic acid as well as stearic acid, that is containing 16 carbon and 18 carbon respectively. Let's move on to unsaturated case. As we have seen, unsaturated means there is going to be a presence of double bond. And for this there is 20 carbon fatty acid, that is arachidoic acid, which is famous for its long chain. Alright, so there is double bond between carbon to carbon. There is formation of kink. Kink is nothing but a structure like this. Whenever I'm talking about a cell membrane, in case of cell membrane, there is one straight chain which is made up of saturated fatty acid, and there is one chain which is pent like this. This is nothing but your site of unsaturation. And this site of unsaturation is basically called as kink. So whenever I am going to use unsaturated fatty acid, there is going to be a formation of kink inside your one structure of your phospholipids. So this is present in every or most of the phospholipid components. Alright. So these are basically liquid at room temperature and they are less stable due to your sigma bond. Alright, so there is less stable, even though there is a presence of sigma bond, along with this sigma bond, there is going to be a pi bond. So due to this pi bond, they are more reactive and less stable. So example for this is oleic acid and linoleic acid. You don't have to remember the site of unsaturation in this particular case, which I have covered in my brief videos about biomolecules. If you want to get information more deeply on this particular concept along with the mechanism of the reaction, you can go and check out my videos on biomolecules. Now, if I'm talking about types of lipid, there are total 3 different types, that is simple lipid, compound lipid and sterol. Whenever there is a simple lipid, that means there is only a presence of fatty acids plus there has to be what? Glycerol. So fatty acid and glycerol, whenever I am going to consider, that is nothing but your simple lipid. For this, there is an example of gingerly oil. Now, there is a speciality of gingerly oil as this remains liquid even at winter. Alright, even though there is a winter condition outside in the surrounding, this oil remains in a liquid state. That is a case of gingerly oil. Otherwise, all fats or lipids become solid instead. If I'm talking about the next case, that is compound lipids. This compound lipids are basically attached with one another group other than fatty acids. So the group can be carbohydrates, the group can be phosphate, the group can be nitrogen containing compound, it can be anything. Alright, so whenever I'm talking about carbohydrate, that is called as cerebroside. Whenever I'm talking about phosphate, that is called as phospholipid. And whenever I'm talking about phosphate group plus nitrogen compound, that is called as lecithin. So in case of lecithin, they belongs to a case of phospholipids only. But if there is more detailed option, that is phospholipid plus nitrogenous compound, then you should take that particular case. And on lecithin, NEET has been asking your questions. Alright, it can be your statement based, it can be direct question, that is in which type of lipids you are going to observe lecithin. It can be your compound lipid or phospholipid. Next case, your sterols. The sterols are basically containing a ring-like structure. Alright, and this constant ring is carried in all of their structure. Maybe it can be your cholesterol, it can be progesterone, it can be testosterone, or used in different anti-fertility pill. So for this, diosgenin and dioscorea are one of the main thing. Diosgenin is a component obtained from this particular plant, that is also called as yam plant. Alright, so dioscorea is called as yam plant and that is going to give me diosgenin, and this diosgenin is used inside your anti-fertility pills. Along with that, the sterols are one of the main components for deciding your cell membrane rigidity, that is cell membrane structure. Then there are some hormones which are made up of sterols. So these are one of the main components of sterol used inside the body of an organism. It can be your plants, it can be your animals. In case of animals, there is going to be hormones and cell membrane. In case of plant, there will be your cell membrane components are rich in sterols. Alright, so let's move to the question. A phosphoglyceride is always made up of. Alright, now phosphoglyceride means there should be a phosphate group. Every of the option should have phosphate group right now. Yeah. So now we are talking about only saturated fatty acid are esterified. Only word is wrong. There is not as such necessity whether I am going to use only saturated fatty acid or only unsaturated fatty acid. Cancel out. A saturated or unsaturated fatty acid esterified to glycerol molecule to which a phosphate group is also attached. This is my correct option. It can be your saturated fatty acid, it can be your unsaturated fatty acid. It should be attached to a molecule called as glycerol, which look like this: CH2OH, CHOH, CH2OH. So on third carbon there is going to be a phosphate group. On 1 and 2 carbon there is going to be a presence of fatty acid. All right. All right. Okay. Let's move on to the next part, that is significance of lipids. Whenever I'm talking about significance of lipid, that means they are one of the main component of cell membrane as phospholipid bilayer. Phospholipid bilayer. Whenever I'm talking about lipids, they act as shock absorber. They surrounds with a layer on visceral organs of body. The organs which are soft, all organs are basically laid by a lipid so that the external pressure should not damage that particular organ. Along with that, they are also present beneath the skin. So by remaining beneath the skin, they are not allowing any heat to pass out from a body. That's why if you are putting a fat person inside a cold place, then that person should not face any difficulties of that particular cold environment because his body is not allowing the heat to pass out from it. All right. While if you are taking a thin person and kept that particular person inside a climatic condition where cold is going to be the main factor, in that case that thin person is going to suffer through a tremendous cold over there. All right. So this lipids are present beneath the skin so that it prevent heat loss is not going to occur. They are also acting as energy source. That means they are providing ATP to the body. Then they are storing unit. These lipid globules are stored in adipose tissues. Now every tissue is comprised of different cells. So adipose tissue is comprised of adipocytes. So these adipocytes are basically storing this particular lipid molecules. Then they are used in research as a hormone. The hormones which can cross cell membrane directly are basically made up of lipids only. That is nothing but your sterol components, and there is going to be a presence inside vitamin D, which is useful for calcium ion absorption. This is nothing but my significance of lipid. Let's move on to the next biomolecule, guys. We don't have to tired in this particular chapter because this is very, very important as per your fundamentals are concerned and as per your NEET is going to be concerned. So as we are talking about proteins, the proteins are made up of monomers called as amino acid, but there should be a presence of charge. Presence of charge occurs during binding of this particular amino acid. So those are called as amino acid residues. So these amino acids are repeated to form a complex structure called as polypeptides, and that polypeptide is formed by peptide bond. I gave you three names currently for every biomolecule. That is, in case of carbohydrate, there was glycosidic bond. In case of fatty acid, there is ester bond. And in case of protein, there is going to be a peptide bond. Now, naturally, there are only 20 amino acid found. Rest, if I am going to get any new amino acids, then all those are basically synthesized inside a lab. And this amino acid are formed after a process of translation. That means DNA is converted into RNA, and this RNA, whenever it is converted into protein, so in this process, amino acids are released, and these amino acids are forming your proteins. Alright, this is a process called as translation. I hope you are understanding this particular part. And here is a list of all 20 amino acids. Alright, let's move to your first question. Which of the following statement is wrong? Cellulose is polysaccharide. We have seen this. This is made up of what? Beta glucose. Uracil is pyrimidine. This we will be covering inside nucleic acid, but this is true. And this is present inside your case of RNA. Then sucrose is disaccharide. Yes, it is made up of your alpha glucose plus beta fructose, and in this case the glycine is sulfur containing amino acid. This is a wrong statement, guys, because the glycine is simplest amino acid, and this is only amino acid with a chiral carbon. Achiral carbon means this carbon is bonded with some of the repeating component, because this structure of amino acid look like this. If I'm talking about glycine, there is C, H, H, COOH and NH2. So in this particular case, the carbon is comprised of two hydrogen molecule, and that's why it is called as a chiral carbon, and there is no sulfur inside this particular amino acid. If I'm talking about sulfur containing amino acid, there are two cases, that is methionine, and there is going to be your cysteine. Alright, the cysteine is used for formation of disulfide bond in the case of protein. Next case, which one out of A to D given below correctly represent the structural formula of basic amino acid? Whenever I'm talking about basic amino acid, that means it should possess positive charge on it. Alright, amino acid means there should be a presence of amine group as well as COOH group present in a structure, and whenever there is a positive charge, there has to be a presence of NH2 group. This you must be knowing right now. Alright, so let's check for a structure. There is COOH group, guys. So this is not your basic, but this is negatively charged, so acidic amino acid. In this case, there is a presence of OH, so this is non-polar. Alright, in this case, this is not even amino acid, as there is no COH group. NH2 fulfill, COH fulfill, H, and there is a presence of NH2. So this option D is nothing but my basic amino acid. Got it? How to understand what is basic amino acid? Alright. The vitamin nicotinamide can be synthesized in our body from. Now this is basically used in NADH and NADPH. Alright, so these are the two components which are present inside animal and plant. Alright, and this acts as my coenzymes. coenzyme. You must have seen this in the case of glycolysis or in the case of photosynthesis as well. So these are formed from tryptophan amino acid. Just let it be in your mind that these are formed from tryptophan amino acid. So let's move to what is amphoteric nature of amino acid. If I'm talking about the normal structure, then your amino acid are comprised of C, then there is NH2, then there is COOH group as amine and acid group. One is hydrogen and one is R group. The R group is changing according to your 20 amino acids. So whenever I'm talking about amphoteric nature, that means two opposite properties are possessed by one amino acid itself. Now, what are these two properties? That means there is going to be your amino acid can act as acidic in nature or it can act as basic in nature along with your zero charge in between them. Let's see what is going to happen. Whenever I'm talking about acid, that means it will release H+. Now, releasing H+ means gaining negative charge on amino acid. So, whenever I am increasing the pH above 7, like I am moving on this side, the amino acid is becoming more and more acidic. Alright, so in basic pH, amino acid act as acid. While whenever I am reducing down the pH below 7, so in this case it will accept H+, to act as your base. It will accept H+ to act as base when in the surrounding the pH is below 7. So there is acidic pH. So in case of acidic pH, amino acid act as a base, while in case of basic pH, amino acid act as your acid. So in this case, this NH2 is accepting one H+ more, so that your positive charge is going to be gained. So whenever amino acid act as basic in nature, it gains your positive charge, and whenever amino acid act as acidic in nature, it will gain negative charge. So in this case, Q is negative, the charge is positive here. While moving to positive to negative charge, there is one state at that pH. At that pH, you are going to see the total charge on amino acid as 0. The total charge on amino acid as 0, and that pH is called as isoelectric point. At this point, the amino acid shows Zwitter ionic form. Now this Zwitter ionic form means the amino acid possesses positive as well as negative charge, but due to the equal magnitude, the complete charge on amino acid becomes 0. Alright, so this is nothing but your amphoteric nature of amino acid. Very, very important whenever we are talking about stability of protein and the working of enzymes. Alright, so this amino acids are joined together by a peptide bond. Alright, so in this peptide bond, you can see the hydroxyl group is coming close to your hydrogen group, and this basically removes out water molecule. This removes out water molecule, and after removing of water molecule, the bond which is formed, that is nothing but C double bond O N H. This bond which is formed, this is nothing but my peptide bond. So for peptide bond, we should know that two amino acids are having one peptide bond. Now tell me, 3 amino acid will have how many peptide bond? 3 amino acids will have 2 peptide bond. Alright. So to calculate number of peptide bond is equal to n minus 1, where n is nothing but number of amino acid. Alright, so this is a formula for your number of peptide bond to find out. Let's move on to the structure of proteins. Whenever I'm talking structures, that means how this particular protein is looking inside a 3-dimensional structure. That is a case. Whenever I'm talking about primary structure, that means there is only a straight chain. The chain of amino acid, and it is least stable. So it is least stable. As we are moving further, this particular proteins are entering inside endoplasmic reticulum, right? In endoplasmic reticulum, there is going to be the folding of this particular straight chain. So that folding helps to form alpha helical structure, and both of this belongs to secondary structure of protein. So alpha helical structure, whenever I'm talking about, in that particular case, there is going to be a presence of hydrogen bond. So this hydrogen bond is forming within a structure itself, and their R groups are exposed on outer side. So this structure is called as alpha helical structure, and it stabilizes itself because of this intramolecular hydrogen bond. Alright, so there is intramolecular hydrogen bond. That means hydrogen bond formed within the structure itself, and that stability along with your disulfide bond somewhere. Alright, so there is a stability is gained due to the folding and coiling of that primary structure. If I'm talking about the secondary structure, that is beta pleated sheet, that beta pleated sheet is basically stacking of primary structure on each other. So in this case, there will be stacking of primary chains on each other to form intermolecular hydrogen bond. So there is going to be the formation of intermolecular hydrogen bond, and this can be folded and used as channels inside a cell membrane. I hope you must be knowing what are channels. Alright, if not, please go and watch my video on the cell membrane. So there is going to be your beta pleated sheath, which are mostly going to act as channels in the cell membrane. Let's move on to the tertiary structure. This is one of the stable structure whenever I am joining more than 2 secondary structure. So if I'm joining 2 or more than 2 secondary structure, then it is going to form my tertiary structure. And similar to that, if I'm joining 2 or more tertiary structure, it is going to form a quaternary structure. This is globular structure of protein and most stable structure of protein. So this is your most stable structure of protein, and hemoglobin is belonging to this particular category, that is quaternary structure. This hemoglobin is comprised of 2 alpha and 2 beta chains. Alright, so there are 2 alpha chains and 2 beta chains which are joined together to form a quaternary structure of hemoglobin. So let's do some questions. Which of the following is least likely? So the keyword is least likely to be involved in stabilizing the 3-dimensional structure. As we have seen, hydrogen bond is going to be there. Some amino acids are basically having charges. So there can be acidic or basic amino acids. So there has to be electrostatic interaction should be there. Then this complete structure of protein is placed inside a cytoplasm. So there is going to be a presence of water, and there are some hydrophobic amino acid. So this hydrophobic amino acid are nothing but basic amino acid. Some basic amino acid which cannot form hydrogen bond. Alright, they are unable to form hydrogen bond. So there are hydrophobic interaction, but ester bond, it is not seen in case of protein. So answer will be option number 4. So this is my answer. That is option number 4. Ester bond is generally seen in case of lipid. So it is seen in lipids in attachment of fatty acid with glycerol. Fatty acid with glycerol. Alright. Let's move to the next part, that is what are the types of protein. If I'm talking about the types of protein, there are 3 basic types, that is nothing but simple proteins, conjugated proteins and derived proteins. If I'm talking about simple protein, they are just made up of amino acid and peptide bonds. The structure can be any from all those 5 structures. It can be your primary, it can be alpha helical, beta pleated sheath, tertiary or quaternary structure. So the structure can be most stable to least stable. Most stable to least stable. But the type is only made up of amino acid. There is no extra additive moieties present inside a structure. So that's why they are called as simple proteins. Next case is conjugated protein. In this particular case, you are going to see a structure of hemoglobin and myoglobin. Along with that, there are some glycoproteins as well. Alright, so antibodies and all are also belonging to this particular category. So conjugated proteins are basically whenever other components such as fats, carbohydrates are basically attached on a protein to form a conjugated structure of 2. Whenever I'm talking about glycoprotein, the glyco means carbohydrates are in less quantity and the proteins are in large quantity. Alright, so major components are protein and minor components are carbohydrate. That is called as my conjugated protein. While if there is some enzymatic action to form, suppose peptones or proteases, so this enzyme, the proteases enzyme is able to form your peptones, and this peptones and peptides are basically some parts of the protein. So in case of derived protein, there is some part of proteins are basically formed. They are proteinaceous in nature, but they are not a complete protein. That's why they are called as derived protein, or there is use of some synthetic amino acid, and this synthetic amino acid is basically like selenocysteine, where in place of sulfur there is selenium element is basically used. Let's move to the question. Genes are packed into bacterial chromosome by. Now most of the students are going to tick on histones, because whenever I'm talking about wrapping of DNA, first thing that comes in your mind whenever you are in the NEET hall, that is histones. But in case of prokaryotes, the histone-like proteins are used. Histone-like proteins are used. So it is not histone, it is just histone-like. So they are basically basic proteins. There has to be a positive charge so that negatively charged gene should be wrapped. So right answer for this question is basic proteins. Let's move on to the next part. Which one of the following is the most abundant protein in animal world? That is collagen, which forms a supportive structure. Alright, it is present in intercellular matrix and supports the complete tissue. Supports the tissue. This is the use of collagen, one of the toughest and most abundant protein in the animal world. Let's move on to the significances of protein, guys. So in the significances, we are going to see there is a presence of collagen, which is one of the important, as we have seen, abundant protein inside the animal world. Then there are some hormones, the hormones which requires cell receptor, for their function. You are going to see that these hormones are mostly in the proteinaceous nature. So for example, you can see insulin, which is made up of only 51 amino acids. Then they are also used inside the channel, transporter, carriers or pump. These all components are present inside your cell membrane to maintain ionic gradient. So ionic gradient is maintained. Then the proteins are also used in the energy source. That means they are going to provide ATP as an enzyme, which is act as catalyst to speed up the reaction. Then they are used in antibody, that is nothing but used in defense mechanism of body. Then muscle growth, that means skeletal muscle development. And finally trypsin, which is nothing but some of the enzyme broke down the protein itself. So these are the enzymes which are proteinaceous in nature and they are able to break the protein. So trypsin is nothing but used for breakdown of proteins. Alright, so these are some of the significances of protein, and this wraps up with the part of protein. If you want to take a break, just go and take 10-minute break for this particular part. Pause this video and then continue for the next part. So moving on to the next biomolecule, that is nothing but your nucleic acid. One of the important case of biomolecule, because this biomolecule is present in the genetic material. Of organism. So if I'm talking about monomers, these monomers are called as nucleotide. And the nucleotide is comprised of ribose sugar plus nitrogenous base plus phosphate group. So these three components are going to make nucleotides, and these nucleotides are arranged in the repeating units to form your polynucleotide structure. Now in this polynucleotide structure, whenever I'm joining all the nucleotide, this is joined by your phosphodiester bond. So there is a phosphate group as mediator. Whenever I'm talking about phosphodiester bond, where phosphate group act as mediator for two esters. We will be seeing that interesting part in the proper structure of phosphodiester bond. Alright. So there is a phosphate group act as mediator for two ester structures. Now this bond is also formed by the removal of water. In the nucleotide, as I've told you, ribose sugar, nitrogen bases and phosphate group is present. The central unit of life that regulates the complete activity of cell on molecular level. So that is nothing but your genetic material. So whenever I'm talking about nucleic acid, most of the nucleic acid is acting as genetic material, and these genetic materials are basically the central unit of life. Alright, this is very, very important as per the molecular biology is concerned. One of the important cases for molecular biology. Then the nucleotide is formed by nucleoside and phosphate group. So if I remove the phosphate group from the nucleotide, the ribose sugar and nitrogenous base only is going to form my nucleoside. Alright, so nucleoside is comprised of ribose sugar and nitrogenous bases, but whenever I'm going to add phosphate group, then it is going to form your nucleotide. This is official short form, guys. It is not from my perspective. Next, if I'm talking about a question, which of the following biomolecule does have phosphodiester bond? As of now, we have seen that nucleic acids are the only cases. So option number 4, take it and move forward. So these are nitrogenous bases. These are nitrogenous bases, all considering nucleic acid. It might be DNA plus RNA. Alright. So if I'm talking about this nitrogenous bases, there are two categories, that is purines and pyrimidids. Whenever I'm talking about purine, they are two ring compound. As we can see, these are two heterocyclic ring. Now, what do you mean by heterocyclic ring? That means, can you see there is a breakage of the ring due to the presence of inorganic molecule, that is nitrogen, right? So such rings are called as heterocyclic rings. But there are ring 1 and ring 2. So that's why the rings are heterocyclic but 2 in number, and those are called as purines. So adenine, which is represented as A, and guanine, which is represented as G, are basically belonging to a category of purines, while whenever there is only a single heterocyclic ring, the particular structure is called to be your pyrimids. So cytosine, which is represented as C, uracil represented as U, and thymine represented as T are belonging to the pyrimidine case. The thymine is specifically used inside DNA. So specific for DNA, and uracil is specific for RNA. So this is specific for RNA. Alright guys, so this was your nitrogenous bases. Let's see which one of the following structure formulae of two organic compound is correctly identified, which is with its related function. So if I'm talking about the first case, that is triglyceride, is it looking like a triglyceride? Are you able to see that? No, right? Whenever I'm talking about this, there is a nitrogen compound, there is a phosphate group, that means it is lecithin. It is lecithin, and this lecithin is basically a component of membrane. So this one is right. And if I'm talking about a structure, then there is, this structure is of adenine. This structure is of adenine. Please go through the structure once so that you will be remembering and there will be no problem in the NEET examination. Alright, so this is adenine. So adenine forms nucleotide of nucleic acid. Both the statements are true. That's why option number 4 is correct. Which of the following pair of nitrogenous bases of nucleic acid is wrongly matched? So for this, you should be knowing that adenine, guanine belongs to purine, and cytosine, uracil, and thymine, AG-cute, is basically belongs to pyrimidine. Alright, so thymine, uracil, pyrimidine, right? Uracil, cytosine is pyrimidine, right? Guanine, adenine, purine, right? Adenine, thymine, purine is wrong. Thymine is pyrimidine and adenine is purine. So this is basically my answer for this question. Let's move on to the structure of nucleotide. This is nothing but your structure of nucleotide. As we have seen, the nucleotide is comprised of ribose sugar, but in this particular case, this pentose sugar. Alright, the pentose sugar is considered to be deoxyribose sugar. Deoxyribose sugar. Now you should be questioning me, how did you come to know that this is deoxyribose sugar? That is on the second prime carbon. On the second prime carbon, I am going to see there is only hydrogen and not hydroxyl group. That means I have removed one oxygen from the 2 prime carbon, and that's why it is called as deoxy. That means one oxygen less inside a ribose sugar. So this is my nitrogenous base, pentose sugar and phosphate group attached. So this complete structure is going to form my nucleotide. Alright. Let's move on to the question. Given below is a diagrammatic representation of one of the categories. Alright. Can you see the phosphate group? No. Can you see pentose sugar? Yes. So there is a pentose sugar. Pentose sugar. Is there a phosphate group? No. And there is X component. Alright. If I'm adding cholesterol, it is not guanine. This is wrong. Amino acid NH2? No. Nucleotide, for this nucleotide we need phosphate group, so no. So the answer should be nucleoside for this question due to absence of phosphate group. Due to absence of phosphate group, the particular answer should be your nucleoside. So let's talk about your first main component of the nucleic acid, that is DNA, which is called as deoxyribonucleic acid. On this deoxyribose nucleic acid, from 2 prime carbon of the sugar, the OH group is removed out. That's why it is called as deoxy. That means one oxygen less. One oxygen is less on 2 prime carbon in the form of hydroxyl group. The stable structure, because nucleotide are arranged in double, is nucleotide arrangement is double in nature. Now what is double? That means DNA is generally double stranded. However, they have exceptions. So except in pi bacteriophage. Alright. So whenever I am considering here phi bacteriophage, that is phi 174. That is phi 174. In that particular case, you are going to see your single-stranded DNA. Single-stranded DNA is going to be seen. This is one of the important. The sugar acts as a backbone of a structure. That means a complete structure. In that case, sugar and nitrogenous bases are very, very important, because sugar is forming a backbone and nitrogenous bases are forming hydrogen bonds. That means adenine is bound to thymine while cytosine is bound to guanine. And these bonds are not ionic, not covalent, not coordinate, but these bonds are basically your hydrogen bonds. So adenine and thymine are bound together by 2 hydrogen bonds, while cytosine and guanine are bound together by 3 hydrogen bonds. So let's move to the structure of DNA. Can you see this particular phosphate group? So there is one ester linkage, alright, and there is a second ester linkage here. So there is 1 and 2 ester with a coordination of phosphate group. That's why it is called as phosphodiester bond, guys. So there is a phosphodiester bond can be seen. But in case of a structure, you are going to see a concept called as polarity. Now, can you see this as 3 prime and 5 prime written? Alright. In this particular case, the 5 prime and 3 primes are nothing but polarity. It can be 5 prime or 3 prime for that particular strand. If the phosphate group is free at 5 prime end, alright, that is 5 prime carbon, then that polarity is called as 5 prime polarity. If OH group is free, then that is called as 3' end. So one strand of DNA is going to have 5' to 3' polarity, while other strand is basically having a polarity just in a reverse direction, that is 3' to 5'. Alright. So now this structure was first identified by Watson and Crick in 1953 as a double helical structure. That means double-stranded DNA was discovered. Now this discovery was first observed actually through a X-ray diffraction pattern by Rosalind Franklin and Maurice Wilkins. These were the two scientists who were expert in the X-ray crystallography technique and X-ray diffraction patterns. So those patterns are basically observed and analyzed by Watson and Crick to understand that the DNA is double helical structure. Now, the structure was seen to be different in prokaryotes and eukaryotes. If I am talking about prokaryotes, it is going to have circular double-stranded DNA, while in case of eukaryotes, there is linear double-stranded DNA. Linear double-stranded DNA. The strands are antiparallel and complementary for proper binding of nitrogenous base. Now, as we have seen, the binding of nitrogenous base needs hydrogen bonds. And for formation of hydrogen bond, your element should be in close vicinity. Elements should be in close vicinity. Now, whenever the elements are in close vicinity, then and then only the hydrogen bond is going to be formed. And this close vicinity will only achieve if we are putting one of the strand in anti-parallel direction than that of your first strand. Now, if I am considering DNA, then there is adenine, thymine, cytosine and guanine are the nitrogenous bases involved, and purine and pyrimidine shows complementary binding. So which were the purines? A and G, and pyrimidines are T and C. So there should be binding of A, double bond T, that is there are 2 hydrogen bonds, and C, triple bond G, that means there are total 3 hydrogen bonds are present. Thus, this particular case that is based on number of purines and pyrimidine is given by Chargaff rule. So this Chargaff rule is basically giving number of purines and pyrimidines. So this was been told that purine is to pyrimidine ratio is always 1. That means A plus G upon T plus C is always going to be 1. That means A plus G is always going to be T plus C. This was the note of your Chargaff rule. The DNA has different polarity. Alright. As we have seen that both the strands run anti-parallel. That means one strand will have a polarity from 5' to 3' and other strand will have polarity from 3' to 5'. Depending on which particular side the OH is free or the phosphate is going to be a free component. Now, the last case, different number of complementary base pairing based on the conditions in surrounding. Now, what does that mean? If we are putting the organism in harsh condition, then C triple bond G content will increase. As it is more stable, there are 3 hydrogen bond and it will require large amount of energy to break that particular bond. So that's why the content will increase, so that the DNA will remain stable. The organism will move towards, the organism's DNA will moves toward gaining stability, guys. So for being stable, it has to increase the number of GC content. So this particular case is called as GC content. Don't confuse if this particular name comes in your question, that is nothing but GC content. All right. So let's see some of the question. If DNA percentage of thymine is 20, what is the percentage of guanine? Now thymine is basically binded with adenine. So all thymine has to be bound with adenine. If thymine is 20%, then adenine is again going to be your 20%. So out of 100%, the 40% is comprised of thymine and adenine, and your cytosine is bound with guanine. So if cytosine is X%, then guanine is again going to be your X%. So 2X is equal to remaining 60%. So X becomes 30%. So guanine and cytosine are basically having 30% contribution inside that particular DNA. In the DNA molecule, let's see, the total amount of purine nucleotide and pyrimidine nucleotide are not always equal. That is not the case. They has to be equal because according to the char graph rule, their ratio is going to be 1:1. Two strands run parallel in 5' to 3'. Wrong. This is shown by char graph. Alright. The portion of adenine in relation to thymine varies in organism. That should not happen because adenine is bound to thymine. So both of them should have same proportion. So that's why adenine and thymine will not have any different proportion. So this is wrong. So obviously the option will be your last one. Two strands which run anti-parallel, one from 5' to 3' and other one is 3' to 5'. Let's move to dimensions of DNA, guys. Dimension of DNA is nothing but first one is diameter 2 nanometer, which is also called as 20 angstrom unit. The diameter is nothing but distance between two strands. And this dimension are specifically talking about bDNA, and which is double strand. Alright. The length of 1 turn of DNA is 3.4 nanometer, which is also 34 angstrom unit. In that particular 1 turn, you are going to see 10 base pairs. Now, what is base pair? That is adenine and thymine, cytosine bound with guanine. This are nothing but my base pair. This is one base pair. This is your one base pair. Alright. So base pair are basically 10. That means number of nucleotide has to be 20. One base pair has 2 nucleotides. So whenever I am considering 10 base pair, that means there are 20 nucleotides are present. The distance between two successive base pair. Now whenever I am talking about the distance between two base pairs, suppose adenine thymine is the first and cytosine guanine is the second. So what is the distance between this? It is nothing but your 0.34 nanometer, which is equal to 3.4 angstrom unit. Then the angle of pitch of 1 turn, from which particular angle the turn is formed. Alright. And this angle has to be in a repeated form in the next case. Whenever I'm talking about 1 turn of the DNA, alright, so this turn is completely made, and from this point it is again repeating as a first turn. Alright, so this complete turn is called as angle of pitch. So the angle of pitch is 360 degree, while the angle between two successive base pair, as there are 10 base pairs, so it will be 360 upon 10. So that is nothing but your 36 degree. And the number of nucleotide in one base pair is nothing but. Alright. Let's move on to the question. 1 ton of helix in B form DNA is approximately 1 ton of helix is 3.4 nanometer. Easy peasy. Now moving on to the next part of nucleic acid, that is nothing but RNA. The RNA is basically called as ribonucleic acid, and it is present with 2' OH group. Now this is the one of the case. It is having 2' carbon with hydroxyl group. This group makes RNA unstable. Makes RNA unstable as it can form phosphodiester bond. It can form phosphodiester bond instead of 3' carbon and make irregularities in the structure. The complete structure which has to be maintained in a regular form where 3' carbon OH group is forming phosphodiester bond with the next nucleotide set. In this particular RNA, the 2' can also get involved. Alright, and the next case will be it forms intramolecular structure. It forms intramolecular structure which stops forming hydrogen bond as well. Alright, so that's why it is mostly unstable structure, and the RNA is a genetic material in acellular entities, such as your viruses. Alright, and this discovery was done by a scientist called as Baltimore. Alright, RNA as genetic material. So let's move to the next part, that is the uracil is placed in the position of thymine complementary to adenine. So whenever we are talking about DNA, in case of DNA, adenine was bounded with thymine, but now in case of RNA, the thymine is replaced by uracil. This is one of the main difference, and the thymine is also called as 5' methyl uracil. Alright, so uracil and thymine will have only one difference in their structure, that is on the 5' carbon there is a presence of methyl group. RNA is obtained from DNA through a process called as transcription. Alright, the process of obtaining RNA from DNA is called as transcription. Some of them are placed inside nucleus and some of them are placed inside cytoplasm. So RNA is generally present in cytoplasm based on their function, obviously. So what are the types of RNA? There is mRNA, rRNA, tRNA, one of the 3 significant types. So these are my 3 significant types which you have to study. And there are 2 more, that is snRNA, that is small nuclear RNA, which silence the expression of gene. That means whenever the gene has to give a product, that product should not be come out for whenever I'm going to use snRNA. And there is miRNA. It's not Mumbai Indians, guys, please. It is basically microRNA. It forms double-stranded structure on RNA and stops gene expression. Alright, so both of them are basically helping to stop gene expression, whether it can be snRNA or whether it can be miRNA. Alright, so let's move on to the first part, that is mRNA. The mRNA is basically your messenger RNA. Now this RNA is comprised of different nucleotides, as you can see, ACC, AU, CG, AU, AG. These are the set of nucleotides. So this is nothing but my nucleotide. Whenever there is a combination of 3 nucleotides, that is called as codon. So codon is also called as triplet of nucleotides, and each codon gives one amino acid. So there is some information is stored in the form of code. All right. So the information for formation of for formation of amino acid is stored in the form of code is basically called as mRNA. So mRNA is going to carry a message in the form of code. That code is helpful for the synthesis of amino acid and finally the protein. All right, and the code is in the form of triplet of nucleotide called as codon. All right, so there are total 64 codons, out of which 61 codons codes for 20 amino acids. Alright, this information is very crucial whenever the molecular biology is concerned. This particular RNA is called as template RNA, also called as messenger RNA. The template RNA because it provides a template for the formation of amino acid, and messenger because it carries a code that is going to help in synthesis of amino acid. Now it carries the information of coding of protein. It is detected by ribosome for protein production. Alright, so it is detected by ribosome for protein production. So there are some conserved sequences inside a ribosome. There are some conserved sequences inside a ribosome which are going to attach on mRNA to check whether mRNA belongs to the same species. That means whether the mRNA is of prokaryotic cell or eukaryotic cell. If prokaryotes is going to have a mRNA of eukaryote, then that mRNA will not be expressed inside the prokaryotic cell, and that role is played by ribosome, which is going to detect this particular mRNA. Alright, clear with this concept? This particular mRNA is very less in number, that is around 3 to 5% of the total RNA content, and it degrades quickly as it is a straight chain single stranded structure. Now, this mRNA is formed within a nucleus. So the DNA to mRNA process in case of eukaryotes is carried out within a nucleus, and then mRNA is released out inside your cytoplasm for protein production. And that protein production process is called as translation. Alright, this is all about mRNA. If I'm talking about rRNA, the rRNA is basically largely present in nucleolus, and it is helpful, and it forms ribosome. That's why it is called as ribosomal RNA. This is the only RNA which can also act as an enzyme. So this RNA can act as enzyme, and even though the polarity is 5' to 3' and the structure is single stranded, even though the RNA is single stranded in structure, this is folded on itself. The RNA is folded on itself, and this helps in gaining stability. So increases stability of the structure. Alright guys, so this is one of the main concept of rRNA. Let's see about the theory part. So rRNA is also called as ribosomal RNA. It forms the structure of ribosome. It is present in nucleolus, which helps in the synthesis of ribosome, and this is high in number. As the ribosome and nucleolus are continuously present in a cell, then this is basically having around 80% contribution of the total RNA content. Now, as the stability of RNA is seen. It is intramolecular interaction, and there is going to be the formation of hydrogen bonds. All right. This RNA shows enzymatic action, which is also called as ribozyme. All right. The enzyme that is in the form of RNA and not in the form of protein, that is called as ribozyme, and this RNA is helpful for the detection of mRNA compatibility, that is through 16S rRNA sequence and 23S rRNA sequence. So for 16S, this is called as Shine-Dalgarno. This is called as Shine-Dalgarno sequence. This is seen in prokaryotes, and there is Kozak sequence, which is seen in eukaryotes. Alright guys, this is all about rRNA. Let's move on to your tRNA concepts. Now tRNA is also called as transfer RNA. Transfer RNA. It is in the form of loop, and these loops are having their own function. If I am talking about D loop, this D loop interacts with enzyme. Now, which enzyme, what enzyme, that you have to study in the molecular biology. All right. So it interacts with enzyme that adds amino acid. Now, on which side the amino acid is going to be added? That is on 3' end. So this 3' end is also called as acceptor end or loading end, where amino acid is loaded, all right. Then there is going to be anticodon. It is opposite to codon, so it detects codon sequence of mRNA for protein synthesis, and there is going to be a presence of T loop that is interacting with large subunit of ribosome. So this interacts with large subunit of ribosome. All right. So there is one more loop, that is variable loop. No one has been understood about it, but still this is scientists think that it helps in deciding the complete structure of tRNA, and this model is called as clover leaf model of tRNA, which explains all the interactions of the tRNA with the surrounding elements. Now, as we are talking about tRNA, the tRNA is the smallest RNA, it is soluble RNA and transfer RNA. All three names are for the tRNA itself. It is smallest, soluble and transfer RNA because it is remain in the solubilized state. All right. It carries amino acids for protein synthesis from 3' end of the structure, which is also called as CCAN. All right. It detects the codon of mRNA and works based on codon detection, that is through anticodon loop, and it is around 10 to 15% of the total RNA. Now, the structure has different loop with specific function, which we have covered earlier in the earlier slide. If you are not remembering it, please go back and check that particular slide again. All right. It is present inside a cytoplasm and remains soluble stage. Now, why it is present inside a cytoplasm? Because it is involved in a process of translation, and this takes place only inside a cytoplasm in presence of ribosome. So the process is taking place in presence of ribosome, and that's why it is basically present inside a cytoplasm itself. Alright, this is all about tRNA. Now the tRNA has two models called as hairpin model and cloverleaf model. The cloverleaf model explained all interactions, while the hairpin model could able to explain only amino acid binding, binding, and codon detection. And codon detection. How the enzyme is interacting, how the ribosome is interacting, how the particular structure of tRNA is maintained, that not has been explained by hairpin model. That's why it was a flop model for tRNA, and the clover leaf model was widely accepted. Now, if we are talking about significance of nucleic acid, which is the last part of nucleic acid, that is, it is acting as genetic material. There is an enzyme formation code. Enzyme formation code means it is in the presence of genes. So based on the presence of gene, there is a formation of enzyme. The regulation of activity is governed, means your protein production or enzymes or channels opening and closing, open and close, everything is governed by your nucleic acid itself. Then there is energy source, that means they are again providing ATP, but only at last stage. If you are not obtaining energy from carbohydrate, protein, fats, then only the nucleic acid starts to break down. They are used in metabolic pathway. So metabolic pathway, there is an indirect role by involving enzyme formation. Then there is a forensic studies, that means your parent detection and criminals, right? That is studied in the forensic study, while there is also research studies inside your molecular biology. Genetically modified organisms are basically formed through this particular studies. And there is a phylogenetics, that means your relations between organisms can be found out by phylogenetics. All right, so this was your complete nucleic acid. Now we are left with two parts, out of which one is major and another is very simple part, guys. So let's start with your main and most important part of this chapter, that is enzymes. The enzymes are proteinaceous in nature, and as we can see, out of all proteins, the enzyme is taking a subset of protein. All right, out of all proteins, the enzyme is taking subset of protein. That's why it is called as all enzymes are protein, but all proteins are not enzymes. All proteins aren't enzymes. All right, so this statements reveal some information that enzyme's nature is proteinaceous. All right, the enzyme is basically act as biocatalyst. That means it is going to speed up the reaction, and now this speeding up is done by reduction in activation energy. Now what is activation energy? That is nothing but the energy given to reactant, the energy given to reactant to reach to its maximum energy stage, which is called as transition state, and after finally formation of transition state, the product is quickly formed, and that is nothing but a speeding up reaction. Now these enzymes are used in the biological system and they are specific for reactants. So this specificity is very, very important. So enzymes are very specific according to their substrate. All right. So let's talk about this question. One of the interesting question asked in the NEET. Which of the following describe the graph correctly? Now, one thing is that the activation energy is this particular energy gap, guys. If I'm talking about the case B, so B should be in absence of enzyme. And A should be in presence of enzyme. as we can see, the activation energy is reduced. Presence of enzyme. So wherever B in presence of enzyme option is there, we have to cut it out. So there is B in presence of enzyme, B in presence of enzyme. Option 3 and 4 are completely wrong. Now, the thing is, we have to take a bet on either option 1 or option 2. So, if the substrate, if the substrate is having higher energy, if the substrate has higher energy than product, this is in the term of energy, then the reaction is exothermic. Then the reaction is exothermic. Wherever there is a delta H, which is this particular gap, whenever the delta H becomes negative, that means it is an exothermic reaction. This is your thermodynamics, guys. So in biology, you don't have to stick to just organism or just biological concept, but there are physics, chemistry and maths also involved. That's why the biology is so versatile and including all the subjects. And after including those fundamental concepts, you are going to get an application out of it. All right. So in this particular case, the reaction is exothermic. We can see the substrate at higher level of energy and product at lower level. So answer will be option number 2. Exothermic reaction, energy, A in presence of enzyme and B in absence of enzyme. Done with this question. A non-proteinaceous enzyme, I gave that to you, that is nothing but your ribozyme, which was obtained by our RNA. So tick and move ahead. Now, if I'm talking about the types of enzyme, there is inactive enzyme, which are also called as zymogens or apoenzymes, and there is active enzyme. So basically, apoenzymes are inactive in nature. But whenever I'm adding some moieties on apoenzyme, then it is going to form holoenzyme. So whenever apoenzyme is added with first prosthetic group, a carbon chain which is non-proteinaceous in nature, which is called as prosthetic group. Next case is cofactor and metallic ion, mostly bivalent cation are used. I hope you are good with your inorganic as well. And there is going to be your coenzymes. So these coenzymes are basically coming from vitamins. So generally vitamin B complex is used for the formation of coenzyme. Whenever all these three groups are attached on the apoenzyme, then the active form of enzyme is formed, which is called as holoenzyme. Alright guys, so these were your types of enzymes. So let's see what are the questions. The questions from enzymes are quietly interesting and you have to be well versed with this topic to solve this particular type of questions. So which one of the following statement is correct with reference to enzyme? So holoenzyme is apoenzyme plus coenzyme. This is my correct answer. The essential chemical component of many coenzymes are, right now I told you that coenzymes are mostly made up of vitamins. An organic substance bound to an enzyme, an organic substance bound to an enzyme, an essential for its activity. Now it is organic substance. The vitamins are also organic in nature because they contain carbon content, right? So bound to an enzyme means it is coenzyme. Even though the prosthetic group is also organic, it is not listed in the complete options, and there is a case of holoenzyme, but the holoenzyme is a type of enzyme, guys. It is not a bound substance. So the bounded substance is going to be your coenzyme. Alright, that's why the coenzyme is correct answer for this question. Let's move to what are the different properties of enzyme. If I'm talking about enzyme, I have told that enzyme is having proteinaceous nature. So whenever I'm talking about protein, I have to be well uh, remembered with the three-dimensional structure of proteins. Alright, so the enzyme should be stable three-dimensional structure. Should be with stable three-dimensional structure. Now, in that three-dimensional structure, the pockets are formed where you are going to see binding of substrate, binding of coenzyme, then binding of metallic ion, etc. Metallic ion, etc. So these components are basically bound in those particular pockets. As the temperature is increasing, at one temperature, activity of enzyme is maximum. That temperature is called as optimum temperature. We are going to see what are effect of temperature and pH on the enzyme, and similarly for the pH, there is a pH at which the enzymes are going to work at a maximum rate, that is called as optimum pH. So these are basically changing according to the organism based on physiological condition. So coming back to biology. So what are the categories of enzyme? There are here 6 different category: O, T, H, oath, L, I, L. Okay? OTH little. You can remember like this. So, and you have to remember this particular category in the same sequence. Alright. So, oxidoreductase, that means they are going to cause a redox reaction. Then there is a transferase, which is helping to transfer a functional group from one substrate to another substrate. So that is basically like a glucose with ATP to give you glucose 6-phosphate. In the presence of enzyme hexokinase, that is again specific for the glucose itself. Then hydrolysis, this is basically whenever your polymers are broken down into monomers. So polymers are broken down into monomers. That means wherever we are removing water to form a bond, the enzyme will utilize water to break that bond again by providing H plus and OH minus to the respective monomers. Then lysis, without using water, whenever we are going to break a particular bond, that is called as your lysis. Alright. Then there is isomerases. Isomerases are basically forming isomer. That means different structural formula, but the same molecular formula for the particular compound. So in this case, the product will have same molecular formula, but different structure. Alright. And finally, ligases are nothing but they are helping to form your phosphodiester bond. And we have seen the phosphodiester bond in nucleic acid. Now, if I'm talking about the first enzyme, alright, this enzyme term was discovered because of identification inside yeast. So, your first enzyme was been identified inside yeast. Alright, just remember this so that if some question asked inside a NEET, you should not be blank during that particular case. Alright, now there are some models of enzyme which are involved. The models of enzyme are basically based on enzyme substrate binding. So these models are formed based on enzyme substrate binding. Alright, there are two models. First one is lock and key model. This enzyme substrate binding is basically whenever enzyme is having one site, this site is called as active site of enzyme or substrate binding site where substrate is bound. Substrate is bound. So there are two model, that is lock and key model and induced fit model in your syllabus. Let's see what are they. If I'm talking about lock and key model, in the lock and key, we can see that key is specific for a lock. The key is specific for a lock where substrate act as key and enzyme act as lock. So in this particular case, the enzyme has to be very, very specifically made. The enzyme has to be made specifically according to substrate. Then and then only this model will work. But if we start to make that particular enzyme specific for substrate, then there are multiple enzymes and cell will only be fulfilled with enzyme and not other elements. So that's why this model was completely flopped. Alright. So in this case, the model was saying that the substrate is acting as a key, it is fixing inside the enzyme, and then the lock is getting opened and the products are formed. So this was your lock and key model. If we are moving to the induced fit, induce means after substrate comes near enzyme, the substrate induces some change. Where in active site, due to which the binding of substrate take place. So in case of induced fit model, the hydrogen bonds formed between substrate and enzymes were very, very important. Even though the substrate shape is not specific with the active site can change its shape according to the substrate. As we can see, there is a square shaped substrate and triangular shape active site, but the shape of active site has been changed due to the induction by substrate present outside the enzyme. So here the enzyme can change the active site shape. All right. Let's move on to the next part, that is some of the question. Transition state structure of the substrate formed during enzymatic reaction is, as I have told you, it is most energetic. It is maximum energetic. So maximum energy cannot be permanent and cannot be stable. So it has to be unstable and transient. Alright, it is for a temporary period of time, that is transient, but it is unstable. Alright guys, let's move on to the effect of some factor on enzyme. If I'm talking about activity of enzyme versus concentration graph, you can see here the name called as Km, that is Michaelis-Menten constant. Alright, this is one of the important keys. Now what is Km? As I am increasing the substrate concentration, the activity of enzyme is increasing till one point. At this particular point, all the enzymes are occupied by substrate. That means if all the enzymes are full by the substrate, then increasing the substrate concentration won't hamper any activity of enzyme. That activity is called as Vmax, that is maximum activity of enzyme. And whenever I'm talking about Km, the Km is nothing but a substrate concentration itself. It is a substrate concentration itself. But whenever the enzyme activity is half of its maximum, at the point when enzyme activity is half of its maximum, then that particular point of substrate concentration is called as Km. Alright. Now this Km value can vary for majority of enzyme. From 10 raised to 5 to 10 raised to minus 2 moles. Alright. This information, if remembered, good. If not, I haven't seen any question directly asked on this particular range. But for you guys not to miss out any of the part, I am going to give this information. Now let's see. The Km is a concentration of substrate at which the activity of enzyme is half of its maximum. As I have already told you, the value of Km decides the affinity of enzyme towards substrate. That means whenever the Km value is less, that means enzyme become active even in less substrate concentration. That means affinity of enzyme towards substrate is more. Alright. So Km is inversely proportional to affinity of enzyme towards substrate. Alright guys. So this is the relation between Km and affinity of enzyme towards substrate. The maximum activity will always be reached when all the enzymes are occupied by substrate. That I have already told you in the graph itself. Alright. Go through this particular part. If there is any doubt, please comment down in the comment box. Let's see some of the question. Km value is dependent on. Have we seen any enzyme concentration or temperature? No. So the Km value is completely dependent on the substrate concentration. Alright. If there is an option called as activity of enzyme or rate of enzyme, then that will be one of the factor, because we were checking substrate concentration at Vmax by 2 for finding out K, that is Michaelis-Menten constant. Alright. Which one of the following statement is incorrect? Nice. From this part, we are going to see one of the difficult questions. A competitive inhibitor reacts irreversibly with enzyme to form enzyme inhibitor complex. Alright. A competitor inhibitor reacts reversibly. Yeah. If I am talking about competitor inhibitor, then in competitive inhibitor, as increasing substrate concentration, alright, this substrate will compete with one of the inhibitor and bind to enzyme. So enzyme substrate complex is more, and this change is reversible. Even though there is a binding of enzyme inhibitor complex, this can revert back to enzyme substrate whenever I am increasing the substrate concentration. So this one is correct, guys. Alright, statement 1 is correct. The statement 2, in competitive inhibition, the inhibitor molecule is not chemically changed by enzyme. Obviously not, because the enzyme is not changing any molecule. Here just an inhibitor is bound to an enzyme. Alright, that's it. The correct thing is there. The competitive inhibitor does not affect the rate of breakdown of enzyme substrate complex. No, they are not affecting. They are just binding to the enzymes so that the enzyme activity will be stopped. But as soon as the enzyme substrate complex is formed, the rate of enzyme will be again the same. So it is not affecting the breakdown of enzyme substrate complex rate. However, the fourth statement is the wrong statement, and this is my answer. As we have asked for the incorrect statement, the presence of competitive inhibitor doesn't decrease Km for enzyme. This is not at all true. Km is not affected by the inhibitor concentration. Alright. Next case. Select the option which is not correct with respect to enzyme action. Alright. You must not have came across this in a book, but you have to be very well versed with this type of question because that can be asked inside a NEET. The substrate binds with enzyme at its active site. Yeah, it is a right statement which is not correct. We have asked. Addition of a lot of succinate does not reverse. Addition of a lot of succinate does not reverse the inhibition of succinic dehydrogenase by malonate. Yeah, this is a right statement because the statement 2 is talking about non-competitive inhibition. Now, if I'm talking about non-competitive inhibition, that is nothing but when inhibitor binds to substrate. When inhibitor binds to substrate, that is its enzyme. So this is basically an irreversible change. Irreversible change. And in this case, whenever the irreversibility has been seen, the enzyme inhibitor complex has to broken down. Alright, there is no other option by which you can revert back the enzyme in its normal function. Alright. A non-competitive inhibitor binds enzyme at an distant site from which the binding of substrate. Yeah, it is true. And because after which it binds to some different side of the substrate, then the enzyme is not open at its active site for the substrate. That's why it is here irreversible change. Whenever the enzyme substrate complex is formed, suppose this is my substrate, this is an enzyme, here I am attaching your inhibitor molecule, then this site is not opening for the substrate and it cannot bind to the enzyme. Alright, so malonate is competitive inhibitor. No, it is, this will be my answer, this is non-competitive inhibitor. An example of competitive inhibition for this is nothing but, if I'm talking about example for competitive inhibition is cytochrome oxidase by cyanide. This will be my answer. Let's move on to the next part, that is concentration of product versus time. If I am concentration of product is seen on the y-axis, this is completely based on enzyme. When no enzyme was used, the reaction was still taking place, but the reaction rate is very, very low. Reaction rate was very, very low. That's why concentration of product will be very, very less. That if I am adding 1x enzyme, then concentration of product is increasing. But however, whenever I am adding 2x enzyme with enough substrate, then product concentration will increase in same time period. If I am talking about 2 minutes of the process, then in 2 minutes, if I am getting 2 moles of the product by 1x enzyme, then I might be getting 4x product in the same time itself. Alright. Then if we are talking about this graph, the enzyme plays an important role in maximum biological chemical reactions. It helps to form a product at a faster rate by acquiring less time. The product formation depends on the presence and absence of enzyme and its concentration used in the process, that we have seen in the last graph. If enzymes use is more, the product formation will be more than in the equal period given for 1x enzyme concentration. That is what I have told you, that in the same time period I am going to get more product if I am using more enzyme. Alright, let's talk about activity of enzyme on y-axis and pH on x-axis. if you are seeing this particular graph, we are seeing that the pH is decreasing. All right, from this to this, the pH is decreasing, and from this point, the pH is increasing. So as the pH is decreasing, that means I am moving to the acidic condition. That means enzyme will act as base, because enzyme is a protein, and due to base, it will gain a positive charge, and its three-dimensional structure is going to be changed. All right, it will open up to your primary structure. All right, and whenever we are moving on the right hand side, here you are going to see acidic pH, or sorry, basic pH. There is going to be your basic pH and acidic nature of enzyme. Acidic nature of enzyme. This will gain negative charge, and this will again disrupt three-dimensional structure is going to be changed. But at one point where maximum activity is seen of that enzyme, that pH, this particular point of pH is called as, here maximum activity is seen, and pH for it is called as optimum pH. And at this particular pH, the enzyme must not be having any charge on it, right? So at this point, there is most of the enzymes with Zwitter ion cases. Alright guys, let's see what is the information. The enzymes are made up of proteins. Thus, the amphoteric nature of amino acids plays a crucial role in this case. Obviously, whenever I am putting the enzyme in acidic pH, it acts as a base, while whenever I am putting the enzyme in basic pH, it acts as acid. So this is nothing but your amphoteric nature of this particular amino acid. When the pH is less, the enzyme acts as base, gains positive charge, but when the pH becomes high, the enzyme becomes negatively charged because of the acidic nature and removal of H plus ions. Alright, the pH at which the enzyme gains its maximum activity is called as optimum pH. However, the enzyme activity is not suddenly dropping down, but it is slowly dropping down whenever the pH is increased. That is because the positive charge or negative charge is not gained at once, but they are added slowly on the structure of enzyme, right? The enzyme activity decreases gradually and not suddenly. Decreases gradually and not sudden in the case of your pH. All right, but if we are talking about temperature, then you can see there is sudden drop in activity of enzyme, and finally the enzyme becomes inactive. All right, so at this particular time the enzyme is degraded. All right, so why is that happening? Because after an optimum temperature, if you are talking about this particular temperature as my optimum temperature, I can see the maximum activity of the enzyme at this particular point. However, if I'm increasing the temperature beyond this particular point, then the peptide bond starts to break down. So beyond optimum temperature, peptide bonds break down, and this is basically degrades the structure of enzyme. And if we are degrading the three-dimensional structure of enzyme, then that particular enzyme will not be of any use. All right, so let's see. The enzymes are made up of protein, thus peptide bonds concept is used in this particular case. However, the proteins are large in number of peptide bonds, and that is formed between amino acids. The enzyme will gain its maximum activity at optimum temperature. However, the temperature beyond will start to break the peptide bond, and that will deteriorate the three-dimensional structure will collapse. All right, that's why the enzyme activity is suddenly dropped. All right, let's move on to your questions. Which of the following statement regarding enzyme inhibition is correct? They have talked about statements, so we can be able to see two statements correct, or more than two, or there can be one, or there can be all. The competitive inhibition is seen when the substrate competes with enzyme for binding to an inhibitor. Alright. Competitive inhibition is seen when the substrate and inhibition compete for active site of enzyme. Right, it is right. The competitive inhibition is seen when substrate competes with an enzyme, not enzyme, it is competing with inhibitor. So, second one is correct. Non-competitive inhibition enzyme can overcome by adding no because it is irreversible, and non-competitive inhibitor often binds irreversibly. So if you are talking about reversible inhibition of enzyme, in this case substrate competes with inhibitor for enzyme's active site. But in case of irreversible inhibition, there is going to be non-competitive inhibition is going to be C. Alright. So this is basically one of the important case where inhibitor do not bind in active site of an enzyme, Alright. This is the NCERT based question, guys. So let's talk about your last part. I hope this is the last part. Yeah, this is a secondary metabolites. So secondary metabolites are such metabolites which are formed by your bacteria or plants, but they are not useful for the organisms. Such metabolites which are formed by organisms and not useful for them and form due to lack of substrate in EDO phase. All of these metabolites are called as secondary metabolites. So there are different categories such as phenols. The phenols are basically containing aromatic ring, that is nothing but a benzene ring present inside it. There are some alkaloids which are basically obtained from the plants. There are nitrogen containing compounds and terpenes which have been obtained from mevalonic acid. So they are obtained from mevalonic acid. So these are some of the types of your secondary metabolites. So if you are talking about at which particular stage the secondary metabolites are going to be obtained, so that is nothing but your EDO phase. Alright. In EDO phase, what is happening? Let's see. As the time is increasing, the concentration is basically the number of organisms. This is nothing but number of organisms, or we are talking about nutrients as well. At time is equal to 0, the nutrients were very high and the organism number was 0. Alright. The nutrient was high and organism was 0 at the number. Alright. So as soon as the nutrient starts to deteriorate down, that means they are decreasing in number, the number of organisms will start increasing. Why? Because all the organisms are using this particular nutrients for their growth. Alright. As soon as the nutrients are over at this particular point, then from this point the stationary phase starts. And in this stationary phase, you are going to see increase in the secondary metabolites. So basically, when the nutrients are completely used by the species, they are undergoing a stationary phase where a cell mass is remaining constant. Where a cell mass remains constant. And at that particular constant phase, you are going to see the formation of secondary metabolites, and that is nothing but your EDO phase. Alright. The growing phase is called as trophophase, while the stationary phase is called as EDO phase. Now, what are the significances of secondary metabolites? There is a morphine which is used in pain relief and cough. In the cough and pain relief, you can see the use of morphine, which is obtained from Papaver somniferum plant. So this is obtained from Papaver somniferum plant. So latex of this particular plant is used for the formation of morphine. Then there are some enzymes which are also obtained as a secondary metabolite, such as cellulase, invertase, lipase. So cellulase, invertase, lipase. These are some of the enzymes which can be obtained. There are organic acids as well, such as gibberellic acid, which is used to induce parthenocarpy in the fruit, that is nothing but seedless fruits. Then there is going to be acetic acid as vinegar. So some of these cases are also coming under your secondary metabolites itself and obtained from microbial organisms. Alright. There are vitamins such as vitamin C, vitamin B12, vitamin B2, vitamin B1. There are various vitamins, that is mostly B complexes, are formed with the help of your organisms again as a secondary metabolite. When I'm talking about antibiotics, there is streptomycin, penicillin, then there is chloramaphenicol. There are various antibiotics. Alright, chloromycin, there is chloromycin. Some of the antibiotics which are also obtained as your secondary metabolites, and this belongs to a category of one interaction between species called as amensalism. Amensalism. Now, what is amensalism? That means one species is neutral, as secondary metabolite is not beneficial for the species, but they are harmful for another species. So this case is called as amensalism. Alright. Whenever the organism is forming antibiotics, these antibiotics are not working against of that particular organism, but they are killing another organisms. Then there is a tannin, which is basically a tea component. That strong flavor of tea is coming because of this particular component, that is tannin, and there is a smell of cabbage that resists a paste infection for that particular cabbage. Alright. So this was a complete biomolecules with all the questions of NEET. If you have any doubt in this particular video, you can comment in the comment box. And if you have any questions more than this particular question types, then you can obviously mention that in the comment or DM us at @loopsofbiology on Instagram, so that I will be able to solve this particular doubt and give you the proper conceptual feedback on this. Alright, please do not forget to like, share and subscribe to our channel, guys, and please share this video among your friends. Thank you, guys. See you in the next video. Bye bye.